Okay, so thank you so much, Allison, for the introduction. And thank you to FAST for having us here again today. It's so nice to see you in person again. And I'm here to provide an update on what we're calling our PTC-AS gene therapy program. So first, who is PTC? For those of you who aren't aware, we at PTC are a global, commercial, diversified biopharmaceutical company with a focus on bringing innovative treatments to those with rare disorders. We were founded in 1998, and at this point, we have over 1,300 employees and a footprint in over 50 countries. I believe when I presented virtually back in 2020, we had about 850 employees, so we're definitely continuing to grow. Our corporate headquarters are in New Jersey, as is our gene therapy manufacturing site, and our international headquarters is located in Dublin. Looking at our pipeline for PTC Therapeutics, you can definitely appreciate that it's very diverse. And we have robust scientific platforms as well as research from splicing molecules and to an oncology platform. But the focus today really is on our gene therapy platform. One thing I did want to point out is for Upstaza, which is not yet approved in the United States, did recently receive or was granted marketing authorization by the European Commission as well as by the MHRA in the UK. So this is actually the first approved disease-modifying treatment for AADC deficiency, as well as the first marketed gene therapy product for direct infusion into the brain. So as you guys can imagine, that we have a lot of learnings in navigating this complex gene therapy space, from preclinical to clinical, regulatory, and now commercial. And we're definitely working to leverage all of those learnings for all of our upcoming gene therapy programs, including the current AS program. So then getting specifically into our gene therapy program in AS, I definitely should have chatted with Jim Wilson beforehand because I know he presented something very similarly earlier today, but we're definitely in alignment. And so the overall takeaway, I won't go through it at the top, but we in our AS program specifically are not yet in first-in-human studies. We are working on those preclinical studies that are first-in-human enabling to support both safety and efficacy. One additional and very important component that I wanted to point out is our CMC development and manufacturing processes. So I know Jim talked a little bit about manufacturing, but definitely want you guys to understand and just want to be transparent about the timing that can take. I think someone in manufacturing mentioned the difference between small molecule manufacturing and gene therapy manufacturing is kind of like making a bike versus a Boeing 747. So definitely a lot more complex, a lot more to take into consideration. It can definitely be a lengthy process. In addition, if you're working with external vendors, you can really add on to that time, trying to get on their schedule. I spoke with someone recently and they were talking like they were about 2.5 years out from having a product for a first-in-human trial. So that is definitely something you guys should be aware of, and it's definitely something to take into consideration. In addition to our tox material manufacturing and first-in-human material manufacturing, then we have our process analytical and our assay development, as well as qualifications of those assays to ensure they're sufficient for use in clinical trials. And then ultimately, this is all to bring a safe product to humans. But sorry I missed this part, but talking about the timing, we did, and this is why at PTC, we have internalized our gene therapy manufacturing processes. So we do have a gene therapy manufacturing site in Hopewell, New Jersey, as I referenced earlier, and I'll talk a little bit about that on the last slide as well. So jumping right into our data, just briefly, for those of you guys who haven't seen our presentations before, this is gene replacement therapy. But we first looked at our PTC-AS vector with a V5 tag to see where it distributed. So first we looked at vector distribution, so DNA, as well as transgene expression, so the RNA. And we saw dose-dependent increases throughout the rodent brain in multiple brain regions. Not as great in the cerebellum, but pretty good overall. And hopefully hopefully it shows up okay for you. It shows up well on my screen, but as Allison has mentioned, look for the red, and that's the UBE3A. And on the right-hand side, you can see in the top right, there's the vehicle-treated animal with no UBE3A staining. And with increasing dose, you can see this good UBE3A staining that's distributed throughout the rodent brain. But this was only one point in time. So we wanted to ensure that this was persistent and stable expression. And we did look in wild-type rats up to 24 weeks post-dose. This is just a representative example in the prefrontal cortex. I shared the DNA as well, but we're really focusing on the protein on the left-hand side. Up to 24 weeks post-dose, we do see both persistent and stable expression of UBE3A. Overall, the takeaway here is that we had widespread distribution seen throughout the rodent brain following PTC-AS administration that is persistent through a minimum of 6 months post-injection in the rodent. So next, knowing that UBE3A is involved in spine development, we also wanted to look at the number of spines coming off of the dendrites. So these little protrusions that you're seeing, the little red rectangles. If you haven't seen this before, this has been published previously in the wild type versus the AS. So you're looking at these spines which are the local focal point of neuronal communication. And again, on the left-hand side, you can definitely appreciate, for Golgi stating that there's a significant increase in the number of spines in the wild type animal compared to the AS animal. They're significantly fewer and they're not as well developed. But with increasing doses of PTC-AS, we did see a significant increase in the number of spines, so overall in the spinogenesis, which is quantified on the right-hand side. So the black bar is the AS vehicle, the white bar is the wild type vehicle, and then with the colored bars, you see increasing doses of PTC-AS. You can definitely see with the higher doses, and actually the highest dose, that it's comparable to wild type levels, so this was recovered. So overall, the take home here is that we had dose-dependent increases in spinogenesis and cortical pyramidal cells, which suggests a potential for improved neuronal communication. So we've got the distribution, improvement in neuronal morphology. Lastly, we really wanted to see, were there phenotypic changes? Were there changes in behavior? Actual changes in neuronal communication? And the answer was a resounding yes. This is a high-level snapshot of our data, not all of our data by any means, but this is both in the AS mouse and the AS rat that we did see improvements across multiple domains. So first, looking at neurologic health and function via the mouse hind limb clasping test, we did see significant improvements there. Then looking at motor function and motor learning via the rotor rod. So we were looking at the comparison of day 1 versus day 2 and wanted to ensure that the mouse is able to stay on the rod even longer than day 1, which we saw in our treated animals and not our untreated animals. And then we also looked at mouse nest building like Jim did as well, and you're looking to see how robust this nest is built. And this is more equivalent to activities of daily living in humans. And ultimately, we saw improvements in recovery of that phenotype as well, especially in the higher doses. Lastly, we looked at synaptic plasticity, so specifically hippocampal LTP or electrophysiology in both the AS mouse and the AS rat. We saw significant improvements across the board, again, especially at higher doses, but at this point we're seeing improvements in multiple domains, and we can only hope that if we can move this forward into first-in-human trials, that we see positive results in humans as well. So finally, where's PTC today in our AS gene therapy program? As I mentioned previously, we're currently doing first-in-human enabling studies and conducting these studies to demonstrate safety and efficacy in support of a first-in-human trial. We're working to refine dose selection, route of administration, as well as the targeted patient population for a first-in-human study. As you guys have heard by others, we definitely want to stay as homogeneous as possible for a first-in-human study, but thinking long-term and other genotypes, I know other presentations have stated for gene replacement therapy that really, from the scientific perspective, it should be very helpful for all genotypes. So this stuff definitely something we're taking into consideration and having active conversations on. We've also established our internal manufacturing processes for the clinical program. So this is the PTC Therapeutics Center of Excellence for Gene Therapy that was established actually in July 2020 in Hopewell, as I mentioned earlier. And this is for the gene therapy development, testing, and manufacturing for clinical trials, and PTC-AS is actually going through this process. Lastly, we're involved in the Angelman Biomarkers and Outcome Measures Alliance. On the steering committee and others are involved as well, and we're definitely working to support those initiatives. So overall, we're working as safely as possible to move forward and try to bring this to a first-in-human clinical trial while continuing to navigate the complex arena of gene therapy. Thank you for your time.